Please explain in detail the genetics of prokaryotic cells, such as bacteria, plasmids, episomes, transformation, conjugation, transduction, etc. The genetics of prokaryotic cells. Prokaryotic cells, such as bacteria, are single-celled organisms that lack a nucleus and other membrane-bound organelles. Their genetic material is organized in a simple and compact manner, which allows for efficient replication and gene expression. This overview will cover the key genetic elements and processes in prokaryotic cells, including plasmids, episomes, transformation, conjugation, and transduction. 1. Bacterial chromosome. The primary genetic material in prokaryotic cells is a circular, double-stranded DNA molecule known as the bacterial chromosome. It is often supercoiled and found in a region called the nucleoid. The bacterial chromosome contains the essential genes for the cell's survival, growth, and reproduction. 2. Plasmids. Plasmids are small, circular, double-stranded DNA molecules that exist independently of the bacterial chromosome. They carry non-essential genes that provide advantageous traits, such as antibiotic resistance, virulence factors, or metabolic capabilities. Plasmids can replicate autonomously and be transferred between bacteria, facilitating the spread of these traits within bacterial populations. 3. Episomes. Episomes are a subset of plasmids that can integrate into the bacterial chromosome. They can exist in either an integrated state or an independent state, depending on the selective pressures acting on the cell. When integrated, episomes can affect the host cell's phenotype by introducing new genes or disrupting existing ones. 4. Transformation. Transformation is a process by which a bacterium takes up free DNA from the environment and incorporates it into its genome. This can occur through the natural competency of some bacteria or through the use of artificial methods, such as electroporation or calcium chloride treatment. Transformation allows bacteria to acquire new genetic information, including advantageous traits like antibiotic resistance or the ability to metabolize new substrates. 5. Conjugation. Conjugation is a form of horizontal gene transfer in which two bacteria exchange genetic material through direct cell-to-cell contact. One bacterium, the donor, contains a special type of plasmid called a conjugative plasmid, e.g., the F plasmid, that allows it to form a pilus, a thin tube-like structure that connects the donor and recipient cells. The conjugative plasmid is then transferred from the donor to the recipient, sometimes along with chromosomal DNA. This process can spread beneficial traits, such as antibiotic resistance, throughout bacterial populations. 6. Transduction. Transduction is another form of horizontal gene transfer that involves the transfer of bacterial DNA through a bacteriophage, a virus that infects bacteria. There are two types of transduction, generalized and specialized. When the bacteriophage infects another bacterium, it injects this DNA fragment, which can then integrate into the recipient's chromosome, transferring new genetic information. Upon induction, the prophage excises itself from the bacterial chromosome, sometimes carrying adjacent bacterial genes. The bacteriophage then packages and transfers this DNA to another bacterium, where it can integrate and alter the recipient's genetic makeup. In conclusion, prokaryotic cells like bacteria possess unique genetic elements and mechanisms for genetic exchange. Plasmids, episomes, and bacterial chromosomes form the basis of their genetic material. Transformation, conjugation, and transduction enable the transfer of genetic information within and between bacterial populations. These processes promote genetic diversity, adaptation, and the spread of advantageous traits, such as antibiotic resistance, within prokaryotic communities.